Okay, it looks like about time to get started. So, pardon me. Okay, so first, let me remind you of what's going on next week. So remember that 1 week from today, October 19th, is going to be test number 2. I will have a study guide out for you on Wednesday. Friday, this coming Friday, is going to be a review session. So please look over the study guide I'll have published on eCampus. Give it a look over by Friday morning. Please come to class with questions ready. I'll be happy to answer any questions, go over anything again on Friday. But again, remember, I'll just give you guys these review sessions as long as people take advantage of them and actually show up, ask questions. So whether you're attending by Zoom or attending in class, please have questions ready, and I'll be happy to answer them the entire period. Also, so because of the exam, that's going to affect our quiz schedule this week. We'll have a quiz on Wednesday, but because there's a review session on Friday, there'll be no quiz on Friday. Also, don't forget to study your quizzes. Again, there's going to be 1 question from each quiz on the exam, so that's an easy way to get some extra points. The exam, just like last time, 40 multiple choice questions worth 2.5 points apiece for a total of 100 points plus a 5-point bonus question. Again, it'll be administered online. So 1 week from today, Monday morning, nobody, whether it's your day to come to class or not, do not come to class, but do, pardon me, do have access to the internet, specifically eCampus. You'll find the quiz, you'll find the test in the same place in the course content section that I had it last time. All right. Also, one thing I forgot to tell you last time is because I'm giving because I'm giving this test online, I do limit the time that's available to you. Last time I forgot, so I gave you the whole period. This time you won't have the whole period. I'll give you 45 seconds per multiple choice question, which is the standard. I mean, there's nothing draconian about that. That's the standard. It's actually plenty of time if you've studied. So you'll have a half hour to complete the test. For most students, that's no problem. And if you studied, that should be plenty of time for you to answer each of the questions. Again, I'll administer the bonus questions separately, and you'll have about 8 minutes to complete that. Okay. I never gave I don't see anybody saying that they can't hear me, so I assume that I didn't do an audio check this morning. If you're attending by Zoom and you can hear me, can you please give a thumbs up or let me know in chat? Yeah, I think I probably would have gotten some comments in chat by now. Good. All right, so actually I forgot to charge my wearable microphone, so we're going with the one in the AV room, the AV equipment that's here today. Okay, so that's about it for announcements. Any comments or questions on what's going on the upcoming week? Okay, let's get back to chapter 7 then. Okay, so chapter 7 is on aerobic respiration, and as you might recall, there are 3 stages to aerobic respiration. Today we're going to start with the 2nd stage, which is the Krebs cycle. Let me get something to write with. So just by way of review, who can tell me what the 3 stages of aerobic respiration are? Okay, so anybody, Zoom or in the class, what's the first stage of aerobic respiration? Glycolysis. Where does glycolysis take place? What part of the cell? Not the chloroplast. That's photosynthesis. Anybody recall? Starts with a C though, so you're close. Cytoplasm. So the first stage is glycolysis, which takes place in the cytoplasm. Now there's an intermediate step between glycolysis and the second stage we're going to talk about today. Sometimes I like to list that as like step 1A, but it usually isn't included in the 3 steps. What comes between glycolysis and the Krebs cycle? So it's got that big long name. We take the pyruvate from glycolysis. Let me see, anybody on the, so I see some people answered cytoplasm. PDHC, coming from the Zoomers. PDHC, or pyruvate dehydrogenase complex. And second step, everybody probably can see it's right up here on the slides. The second stage is the Krebs cycle. Krebs cycle is also known as the citric acid cycle. I think your book uses the Krebs cycle, so we're going to stick with Krebs cycle. It's also sometimes referred to as the tricarboxylic acid cycle. And you might be thinking, well, why does it have all these different names? It turns out the guy Krebs, the guy who it's named after, is a scientist who wasn't particularly well liked. So people don't like to, this is a very important metabolic pathway. So a lot of people don't like to call it the Krebs cycle because this guy was not particularly well liked, but it's stuck. We still call it the Krebs cycle. Sometimes the citric acid cycle, sometimes the tricarboxylic acid cycle, but let's just stick with the Krebs cycle for now. Okay, so now how about the third stage? Anybody in class or on Zoom? ETC. Somebody from Zoom has typed in. ETC, or electron transport chain. We also refer to this as oxidative phosphorylation. Okay, and you might have noticed that I've been keeping track of all of the reduced cofactors. So we get 2 NAD reduced to NADH in glycolysis. We get, for each glucose molecule, we get 2 NAD reduced to NADH in the pyruvate dehydrogenase complex. Remember these are going to come in really, really important. They're going to become very important in the third stage. Okay, electron transport chain, or oxidative phosphorylation. Okay, so the second stage is the Krebs cycle. PDHC, the Krebs cycle, and oxidative phosphorylation, or the electron transport chain, all of these take place in the mitochondria. All right, so glycolysis, cytoplasm, PDHC, Krebs cycle, and ETC all take place in the cytoplasm. So in the Krebs cycle, acetyl-CoA enters the cycle as a 2-carbon molecule. So remember, in PDHC, we had to prepare the 3-carbon pyruvates to enter the Krebs cycle. So in PDHC, we take the 3-carbon pyruvate, convert it into a 2-carbon acetyl-CoA. Acetyl-CoA enters the cycle as a 2-carbon molecule and forms a bond with a 4-carbon molecule called oxalo acetate. The Krebs cycle produces 3 NADH, 1 FADH2, and 1 ATP. So in the process, 3 NAD are reduced to NADH, 2 FAD are reduced to FADH2, and we get 1 ATP by substrate level phosphorylation. Okay? And there's a question here, how many per glucose molecule? So can anybody think about that? So each time a glucose molecule enters, each time a glucose molecule goes through aerobic respiration, how many NADH, how many FADH2, and how many ATP do we get from the Krebs cycle? Anybody think about that? So remember when we talked about the pyruvate dehydrogenase complex. For each glucose, so each time a pyruvate goes through PDHC, we get 1 NAD reduced to NADH, right? But each time a glucose goes through glycolysis and then PDHC, we get 2 NADH, right? Because glucose is split into 2 pyruvate. Each time one of those pyruvates goes through PDHC, we get an NAD reduced to NADH, so we get 2 NADH per glucose molecule. Okay? So knowing that after we go through PDHC, we get 2 acetyl-CoA for every glucose molecule, how many NADH, how many FADH2, how many ATP are we going to get per glucose molecule? Anybody on, I don't see anybody on Zoom answering. Anybody in class? You're probably overthinking this. This is actually a really simple question. Okay? Double. Yeah, somebody, Ian on chat says double. So yeah, because the glucose molecule is split into 2 pyruvate, we get double the amount of NADH from PDHC, from the 2 pyruvate going through PDHC. So from PDHC, we get 2 acetyl-CoA per glucose molecule. Each time an acetyl-CoA goes through, we get 3 NAD. So that's 6 NAD total. Okay. Each time 1 acetyl-CoA goes through, we get 1 FADH2. So 2 FADH2 total. Okay, can everybody see that? Okay, remember, this is important because we need to keep track of all these reduced cofactors. At the end, we're going to total up all of the ATP that we get from these cofactors and from substrate level phosphorylation. So we need to do some accounting. We need to keep track of this stuff. Okay. Last point here. At the end of the cycle, a 4-carbon oxyl acetate molecule is regenerated. The Krebs cycle is a cycle, okay, just like the Calvin-Benson cycle or the light independent reactions of photosynthesis. So remember, cycles are a type of metabolic pathway where we regenerate one of the molecules that we start with at the end of the pathway. So just like with the Calvin-Benson cycle, we regenerate a 5-carbon molecule of RUBP at the end. In the Krebs cycle, we regenerate a 4-carbon molecule of oxyl acetate. Okay? So if you remember the points on this slide, actually there's one more thing. Okay, I guess that's not that important. If you remember what's on this slide, you should be good for the test. Okay, so actually let me write it down anyway. Okay, so those of you who are on Zoom, I'm actually going to write on the board. I just put up this blank notepad document just so I have a nice white background to write on. Okay, so the Krebs cycle. Okay, so at the beginning of the Krebs cycle, we're starting with a molecule, a 2-carbon molecule of acetyl-CoA and a 4-carbon molecule called oxaloacetate. So we start with okay, so next, in the process of the Krebs cycle, we're going to reduce 3 NAD to NADH. We're going to reduce 1 FAD to FADH2. We're going to make 1 ATP by substrate level phosphorylation. And also, another thing that I want to include is we're actually going to lose a carbon. We're going to make a carbon dioxide, just like we made carbon dioxide in PDHC. So we're actually going to lose 2 carbons. We're going to make 2 carbon dioxides. So in the process, three NAD are reduced to NADH. 2 FAD are reduced to one FAD is reduced to FADH2. One ATP is made by substrate level phosphorylation. And 2 CO2 are produced. Okay. And in the end, we regenerate that 4-carbon oxaloacetate so it can go back into the cycle. Okay, can you guys see that? I'm going to zoom in a little bit to make it easier for the Zoomers to see. Oops, that's awful touchy. Okay, so there, I'll get out of the way so you guys can see that a little bit better. So if you remember these things for the upcoming test 1 week from today, you'll be good as far as the Krebs cycle goes. So we start with the 2-carbon acetyl-CoA. Of course, we got that from PDHC, and a 4-carbon oxaloacetate. In the process of the Krebs cycle, we're going to reduce 3 NAD to NADH. We're going to reduce 1 FAD to FADH2, and we're going to make 1 ATP by substrate level phosphorylation. And we also lose 2 carbons as CO2 in the process of the Krebs cycle. At the end, the 4-carbon molecule of oxaloacetate is regenerated. All right, I'll just give you a second more with that. Actually, I'll make sure that I post this lecture on a link to this lecture on eCampus. So if you missed the notes, you can go back and get what I wrote off the board off of the YouTube link. Okay, let's go back to the PowerPoint. Okay, so if you remember the things on this slide, the things that I wrote down on the board, you should be good for the upcoming exam. Okay. So here's just a diagram of the Krebs cycle. Again, let me go through what I said before. So we're going to start with, in this diagram, each of the red balls represent carbons. So we start with a 4-carbon oxaloacetate. We're going to react that with a 2-carbon acetyl-CoA. In the process, 1, 2, 3 NADs are going to be reduced to NADH. 1 FAD is going to be reduced to FADH2. We're going to make 1 ATP by substrate level phosphorylation. And here and here, we're going to produce 2 CO2. Okay, we're going to lose 2 carbons as CO2. Okay, so I think that about does it for the Krebs cycle. All right, any questions on the Krebs cycle before we start talking about the 3rd stage of aerobic respiration? I don't see any questions from Zoom. Okay, so oxidative phosphorylation, otherwise known as the electron transport chain, or ETC, is the 3rd stage of aerobic respiration. In the electron transport chain, NAD and FADH2 transport their high energy electrons to an electron transport chain embedded in the inner mitochondrial membrane. Okay, so just by way of review, so remember that a mitochondrion has 2 membranes, right? It has a smooth outer membrane and it has a highly folded or convoluted inner membrane. So if I were to try to, hopefully I can draw with the mouse today. So if we have a mitochondrion here, so here's the smooth outer membrane, and the inner membrane contains all of these folds. Kind of like that. It's not too bad for drawing with a mouse. So the electron transport chain is located in the inner membrane of the mitochondrion. So right here, okay, the inner membrane. FADH2 enters the electron transport chain at a later point than NADH. 3 components of the chain are proton pumps that pump protons into the outer mitochondrial compartment. Okay, so in this case, the proton pumps are going to pump protons from here to here. Okay, so in other words, we're going to, again, just like with photosynthesis, we're going to use the energy from electrons going to lower energy states to power proton pumps. These proton pumps are going to build up a proton concentration gradient. Okay, because we're pumping from here to here, that means we're going to have a high concentration of protons here. Okay, that's really awful. That's supposed to be a proton in square brackets with an arrow indicating high proton concentration. The high proton concentration is going to be here. Low proton concentration well, that's not too much better. Okay, but again, this is supposed to be a H plus symbol for a proton in square brackets indicating concentration. So low proton concentration is going to be here in the inner mitochondrial compartment. Okay, So again, we're making a proton concentration gradient that we can use to make ATP. Right? And this point that FAD enters at the electron transport chain at a later point, this will become important a little bit later. Let me just erase this stuff. Okay, so here's the electron transport chain. So this right here is the inner membrane of the mitochondrion. The mitochondrial matrix, this is the inner mitochondrial space. So this is inside, or the lumen of the inner membrane of the mitochondrion. That's here, okay? This is the outer mitochondrial compartment. So this is the space between the outer membrane of the mitochondria and the inner membrane of the mitochondria. And this is the electron transport chain, okay? So this is very similar to the electron transport chains that we saw in the light-dependent reactions of photosynthesis, okay? So each time that a proton gets passed from, I'm sorry, I'm thinking the proton concentration gradient, electrons. Each time an electron is passed from one component to the next in the electron transport chain, it's going to go to a lower and lower energy state. Okay, again, you can think of this like the steps on a set of stairs. So each time an electron is passed from here to here to here to here, etc., it goes down an energy level, just like going down a set of stairs. Okay. Again, just like with photosynthesis, some of the energy from electrons going to lower energy states can be captured and used to do work in the cell. Specifically, with this electron transport chain, the electrons are going to power 3 proton pumps. Okay, so look here. So here's 1 proton pump here, 1 proton pump here, and 1 proton pump here. Okay? The flow of protons through ATP synthases embedded in the inner mitochondrial membrane back into the inner mitochondrial compartment drives ATP production. So this molecule over here, this is ATP synthase. Okay? We've seen ATP synthase before in the light-dependent reactions of photosynthesis. So remember that in photosynthesis, we have protons trapped in the thylakoid compartment at a high concentration. They want to go down their concentration gradient through the channel in ATP synthase, and some of the energy of them going down the concentration gradient is captured and used to drive ATP synthase. Okay? Does the exact same thing here. Okay, only in this case, the high proton concentration gradient is going to be in the outer mitochondrial compartment here. Oh, it's easier to draw big. There we go. Okay, high proton concentration here. Okay, this is from all of the proton pumps pumping protons into the outer mitochondrial compartment. Okay, we're going to have a low proton concentration here in the mitochondrial matrix or the inner mitochondrial compartment, whichever you prefer to call it. Low proton concentration here. Okay, so of course protons are going to want to flow down their concentration gradient. Okay, they can't diffuse directly across the phospholipid bilayer of the inner membrane, so they're going to diffuse through this pore in ATP synthase down their concentration gradient. Just like with the light-dependent reactions, some of the energy of protons going down the concentration gradient is going to be captured and used to drive the synthesis of ATP from ADP and inorganic phosphate. Okay, so we see that, well, they didn't draw, yeah, here it is. So ADP plus inorganic phosphate is going to be used to make ATP driven by the energy of these protons going down their concentration gradient. Is everybody with me so far? So Very similar to what happens to the way ATP is made in the light-dependent reactions of photosynthesis. Oops, I meant to be erasing, sorry. Eraser. There we go. Okay, so now remember a couple slides ago, I told you that FADH2, the electrons from FADH2 enter the electron transport chain at a later point than the electrons from NAD. This is where it becomes important. So NAD pumps 3 protons across the mitochondrial membrane as they pass through the electron transport chain. They will be used to make 3 ATP. FADH2 pumps 2 protons across the mitochondrial membrane as it passes through the ETC. They will be used to make 2 ATP. Okay. So over here, look where NAD is donating its electrons to the electron transport chain. That's over here. Okay. FADH2 carries electrons that are in a little bit of a lower energy state than those of NADH. So they enter at a point that bypasses the first proton pump. Okay, so they're going to enter at a point somewhere between the first proton pump and the second proton pump. So this is where FADH2 enters, or the electrons from FADH2, I should say, rather. Okay, so because NAD donates its electrons at a point where it can power all 3 proton pumps, that means for each NADH that donates electrons, we get 3 protons pumped from the mitochondrial matrix into the outer mitochondrial compartment. Because FADH2 enters at a place in the electron transport chain that bypasses that first proton pump, only 2 protons are going to be pumped from the mitochondrial matrix into the outer mitochondrial compartment per FADH2. Okay? So for the time being, ignore these coefficients in front of the protons. Okay? So now, each time a proton goes down its concentration gradient through ATP synthase, that's going to provide the energy to make 1 ATP. Okay? So, you guys are going to have to think a little bit about this. Knowing that for each NAD, for each NADH, 3 protons are pumped across the membrane into the outer mitochondrial compartment, and knowing that each time a proton goes down the channel in ATP synthase and ATP is made, how many ATP are we going to get for each NADH that gives up its electrons to the electron transport chain? Okay, so it pumps 3 protons out of the matrix. So if somebody's asking how many, 3. Okay, so again, Ian answered 3. That's right. So because NAD pumps 3 protons out, when those protons go back down the concentration gradient, we get 3 ATP. So each NAD that gives up its electrons to the electron transport chain, for each NAD we get 3 ATP. Oh, again, I apologize. I'm not good at drawing with a mouse. 3 ATP per NADH. Okay, and because the electrons from FADH2 bypass that first proton pump, only 2 are pumped out. when they go back down their concentration gradient, we get 2 ATP per FADH2. Whoa. I know there are actually people out there who can do amazing artwork with a mouse, but I can't even spell. Okay, so 3 ATP per NADH, 2 ATP per FADH2. All right. Okay, so in order to keep these electrons moving through the electron transport chain, we need a final electron acceptor at the end to take those electrons away. So here's a way to think about it. So imagine that I have a short piece of garden hose, and then I have a bunch of marbles. So I'm going to take the marbles and I'm going to stuff them into the piece of garden hose, and I'm going to hold my thumb over the other end of the short piece of hose so that the marbles can't fall out. Okay? Eventually, I'm going to fill up the garden hose, and I won't be able to push any more marbles in. Okay? That's basically what the electron transport chain is like. In order to get marbles in one end, I'm going to have to take some out the other end. So we need an electron acceptor, okay, to take the electrons out, just like I need to remove my thumb to let some marbles out to get marbles in to keep them moving through the piece of hose. Okay? So we need a final electron acceptor to take electrons away so that we can keep electrons moving through the electron transport chain. We do this with oxygen. Okay? Oxygen is the final electron acceptor. So we take 2 electrons plus 2 protons plus 1/2 of an O2 molecule, okay? And that makes water. This might sound familiar to you. This is basically the reverse. So remember, in the light-dependent reactions, we have to split a molecule of water to get replacement electrons for P680. Here we're doing the opposite. Okay, we're taking oxygen and we're making water rather than splitting water for electrons. Okay, so, again, let me try to write with this. So we're going to take 2 electrons. Two electrons plus 2 protons. Plus 1/2 of an O2 molecule, okay, which is basically just an oxygen atom. Just to make this look clear, we usually refer to an oxygen atom in biology as 1/2 of an O2. So 2 electrons plus 2 protons plus 1/2 of an O2 gives us H2O, water. Okay? So this is very important. So oxygen is the final electron acceptor. Again, it's important because we need to keep electrons moving through this electron transport chain. In order to do that, we need something to take electrons out of the end. Okay, and that's done. So right down here, O2 plus 2 electrons plus 2 protons is going to give us water. Okay? Is everybody okay with that so far? Everybody understand? Good. All right, so I told you guys before, I think I'm going to write on the board for this. We're going to keep track of all the reduced cofactors, all the ATPs that we made from substrate level phosphorylation, and we're going to do a little bit of accounting. So we're going to figure out exactly how many ATP we can make by completely metabolizing 1 molecule of glucose in bioaerobic respiration, rather. Okay, so let's go back. Okay, again, for those of you zooming in today, I'm going to just put up the blank notepad. Again, this is so I have a white background to write on the board. And let me make sure you guys can see the board well. How am I doing on time? Oh. All right, I'm going to have to be kind of quick about this. All right. Let's look at so glycolysis. PDHC. Oops. PDHC. And Krebs. And let's look at ATP from substrate level phosphorylation. NADH. And FADH2. Okay. Does anybody remember how many ATP we get from substrate level phosphorylation in glycolysis net? 2. Good. Anybody remember how many NAD are reduced NADH in glycolysis? Nobody? I don't see anybody on Zoom. We're running low on time, so I'm just going to go ahead and tell you 2. Okay, and we don't get any FAD reduced to FADH2. PDHC, so we don't make any ATP in PDHC. Per glucose molecule, remember that we reduce 2 NAD to NADH and 0 FADH2. In the Krebs cycle, does anybody recall how many ATP we get per glucose molecule? 1 per acetyl-CoA. How many per glucose molecule? 2. How many? I think somebody answered NAD. How many NAD? 3. Well, per glucose molecule? Per glucose molecule, 6. Exactly. How many FADH2 per glucose molecule? 2. Okay. So that's 4 ATP from substrate level phosphorylation, 10 NAD, 2 FADH2. How many ATP are 10 NADH worth in the electron transport chain? So remember, we get 3 ATP per NADH. So how many ATP are we going to get from 10 NADH? 30. Okay, so for FADH2, we're going to get 2 per each FADH2. So how many ATP are these 2 FADH2 worth? 2 times 2 is 4. 4 ATP. Okay. So 4 ATP plus 30 plus 4 equals 38 ATP. And I know I'm running low on time here. Yeah. So how many ATP have I been telling you guys that we get from metabolizing 1 molecule of glucose? Anybody recall? I don't see anybody chiming in on Zoom. I've been telling you 36, right? Okay. And I'll tell you what, we didn't make a mistake here. There's a reason for this, but we're out of time. We'll talk about this on Wednesday. Remember, test on Monday, quiz on Wednesday, and I'll shut up because I'm running out of time. So unless there's any questions from anybody in class or on Zoom, I'm going to stop the meeting, and I'll see you guys Wednesday on Zoom or in class. So long, guys.